Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to see another biochemical test that is related to phenol red fermentation test. If you guys remember, we already talked 20 biochemical test and it is present into my channel. There will be also a link in the description box that will be the biochemical test, so you can check all the biochemical tests, that how to perform the biochemical test. I already explained all the biochemical test. Okay, now this is another video lecture that is also related to biochemical test, and in this video we are going to talk about phenol red fermentation test. Now what is phenol red fermentation test? As the name suggests, so this is actually the test in which we find out mostly enteric bacilli, gram-negative enteric bacilli, that are the enterobacteriaceae. Okay, enterobacteriaceae like E. coli, like Klebsiella, like Shigella, like Salmonella, like Cetrobacter. Okay, now in this test, what's going on? In this test, we add a carbohydrate, that is glucose, lactose, maltose, mannitol or sucrose. Okay, when we add any of the carbohydrate to the phenol red broth, we have already phenol red broth, commercially it is available, and when we add the carbohydrate, so what will happen? The bacteria will utilize the carbohydrate. Okay, when the bacteria utilize the carbohydrate, it will produce acid, and due to this production of acid, what will happen? The phenol red, that is the indicator, pH indicator, it will turn to yellow, and this indicate that the bacteria utilizing the ferment the carbohydrate and produce the byproducts, and due to this way the color is changed from red to yellow. This is how the whole process is working. Moreover, sometimes the bacteria will produce gases that will be detected by this test. So this is how the test, and this is how the test is working. Now in this test, as I already told you, that we have already phenol red media, but with this phenol red media we add glucose or lactose or maltose or mannitol or sucrose. You can use any one of the carbohydrate while performing that test. The objective means the purpose why this test is performed. So this test is specially used to check the fermentation of the carbohydrates and the gas production, okay, to differentiate gram-negative bacilli. Now what we required while performing that test, so in this test we required personal protective equipments like goggles, gloves, overall, cap, each and everything. Next we need phenol red broth media. Okay, next we need carbohydrates, any one of the carbohydrates like glucose, lactose, maltose, sucrose. Okay, next we need Durham tubes, test tubes, fresh bacterial culture, autoclave, incubator, spirit lamp and burner. So these are the main things that we required in this experiment. The principle means how the whole process is working. Again in this steps, again in this case, what's going on? Let's suppose we have prepared the media, phenol red, we have prepared the media, and we inoculate the bacteria, okay, in this media. Once the bacteria is inoculated, then what will happen? There are some bacteria that will utilize and ferment, we can say ferment the carbohydrate like glucose, and let's suppose if they have ferment the glucose, so then it will be converted into pyruvate during glycolysis. Some bacteria using alternative pathway. Okay, so let's suppose if they are using bacteria are using glucose and convert it into pyruvate, what will happen? The media that is red in color, which phenol red, that is a pH indicator, so it will turn into yellow because of the acid production. Okay, pyruvic acid means pyruvate means pyruvic acid. So due to the production of the acid, what will happen? The media is changed, the media color is changed from red to yellow. First thing. Second thing is that what will happen? Sometimes the bacteria will produce gases, and that type of the gases will be detected in a Durham tube. I will show you the test in next video, in the next slides. I will show you the picture in the next slide, the result. Okay, so when the bacteria produces the gases, so it will be detected and trapped into the Durham tubes. So this is how the whole process is working. If we talked about the test method or the procedure, it's very simple. First of all, you need to sterilize the phenol red broth by the addition of 4 carbohydrates, and then you will sterilize it. Once the sterilization is done, then you have to put these media into the broth. Then you have to put this broth into the test tube, and then you will put a clean sterilized Durham tubes, and then you will inoculate your bacteria, fresh bacteria. After that, you will incubate the tubes for 24 hours, and after 24-hour incubation, you will check the results. How the result means that you will check the color, okay, as well as you will check the gas formation. Now this is how the whole process is working. The purpose means on what purpose we use this test. This test is used to as we talked in the objective, so this test is used to determine the carbohydrate fermentation, to differentiate different types of the bacteria, especially we have Enterobacteriaceae, like we have gram-negative bacilli, like we have E. coli, Klebsiella, Salmonella, Enterobacter, Citrobacter, etc. So these types of the bacteria we are identifying through this test. This is the result, as you can see. So this is the negative result. Now this is the Durham tubes. This second one is the Durham tube. This is the media, and after 24-hour, what we get, if we get this type of the result, it means it is negative. If we get this type of the result, it means it is positive reaction. Okay, and let's suppose if we get this type of the, it means it is also positive, but this space into the tube indicating the production of the gases. Okay, so this is how the whole process is working, and this is the result of the phenol red fermentation test. I hope this video is helpful for you. If you like this video, make sure to hit the like button and share this video with your friend. Thank you so much for watching.